Hello, everyone. Thank you so much for joining us today for the first episode of the TNE Seminar Series. That is a name that's in progress in terms of what it's called, but essentially the seminar series is about ensuring that we give researchers in the African diaspora the opportunity to speak about their area of expertise. And so for this episode, we will be discussing immunostaining sample preparation. Nehal be talking about that for us. Nehal is from in neuroscience, an Egyptian neuroscientist who's pursuing her Ph.D. at the American University in Cairo and is a research assistant at the Zewail Science and Technology. And we look forward to Nehal teaching us about sample preparation today. We will have a 5-minute Q&A session after Nehal's presentation where you guys are free to raise your hand so we can unmute you for a question or post your questions in the discussion box. And so without further ado, Nehal, please.
Hello everyone. Thanks Precious for this introduction. I will share my presentation now.
Yes, you can share.
Hello everyone, I am Nehal Ghoneim. I'm a Ph.D. candidate at the American University in Cairo, research assistant and instructor at the Zewail City of Science and Technology. At first, thanks for African Bioimaging Consortium for hosting me today to give my lecture about immunostaining sample preparation in life sciences. And I would encourage you to join African Bioimaging Consortium and follow up on the newsletters to join the future workshops, seminars, and webinars by the ABIC community. Okay. At first, let's have a look about the history of immunostaining and the immune fluorescence staining. In early 1920s, Michael Heidelberger, the father of modern immunology, was the first to use the dye to label antigen. So he was the first one visualized antigen-antibody reaction using labeled antibody to detect the pneumococcal polysaccharides. And after then, in 1934, Professor Marak described the lattice formation of antigen-antibody complexes. And then in early 1940s, the Professor Kohns was the first person to conceptualize and develop immunofluorescent technique for labeling the antibody. Actually, he is the father of immunofluorescence staining. So I would like to share with you the quote about immune fluorescence by Professor Kohns. He is saying that fluorescent antibodies, whatever their scientific merits, are very attractive under microscopes. They shine in the dark, a brilliant greenish yellow glow, like pupils in the moonlight. They weave in a pattern in the forest, which leads the weary children home. What a beautiful quote, actually. Okay, we know that to detect a protein inside the cell or on the surface of the cell, there is a lot of techniques. There are many powerful techniques used to study structure and function of proteins. Antibodies are specific and secreted against specific proteins that usually bind to the surface of the protein. Among these various lab techniques, enzyme-linked immunoassay, Western blot, immune precipitation, immunohistochemistry, flow cytometry, and our target today, which is immunocytochemistry. So, what is the importance of protein analysis and the importance of immune staining technique in a specific, or immunocytochemistry technique in a specific? Protein analysis is fundamental to understand the human physiology and is useful for diagnosis and guiding treatment and even monitoring human diseases. During the basic science research to evaluate the location and co-localization of proteins within the cells, for instance, in nucleus or in cytoplasm or on the membrane, immune staining is used for this. So if you wanted to co-localize the protein, one of the important technique, actually it is the most important technique, is to use the immune staining or immune cytochemistry to co-localize the protein. As we can see in the figure, the protein A is green fluorescent and protein 2 is red. So we can co-localize them that they are overlapped or they are beside, or we can, and also in the figure below. Also here we can see and determine if our protein, which is in green is nuclear or extranuclear in the cytoplasm or even on the surface of the cell membrane. So the localization can be determined using the immune staining. Immune cytochemistry, which is the proper term or the general term of immune staining, is used to study the subcellular distribution of the protein using labeled antibodies. It is a technique that uses antibodies. That's why it's immuno in cells cytochemistry, because antigen antibody reaction is used to visualize the protein of interest. In contrast to immunohistochemistry, that the focus on the samples of tissues or tissue blocks. Here, in cytochemistry, we focus on the cells, not tissue blocks. Okay, the antigen-antibody reaction is visualized using different biomarkers. Different markers. Common markers include fluorescent dye, enzymes, colloidal gold, or even radioactive elements. So what is the meaning of antibodies used? Primary antibody, secondary antibody. What is the difference? These are antibodies used. Primary antibodies is the antibodies that bind specifically to that antigen of interest or to your target. And the secondary antibodies are the antibodies that react or they bind to the primary antibody and not to the direct and not directly to the target of interest, the protein of interest. Okay, so how it is helpful, how this information is helpful, because there are two different techniques: direct immune cytochemistry or indirect. The direct one uses the conjugated antibody, which is the primary antibody conjugated to our fluorescent dye, as in the left side of the image, that bind directly to the epitope of antigen or the protein of interest. However, in comparison, on the indirect immune cytochemistry, unconjugated primary antibody will bind to the antigen, and then it will be detected using secondary antibody, which will react to the primary antibody and bind to it. And this secondary antibody, actually, it is labeled using a fluorescent dye. So it is either the primary antibody directly conjugated to our fluorescent dye and bind to the protein of interest, or it is in like a sandwich technique. It's between the secondary antibody and our protein of interest. So what are the major steps or major components in immune staining? At first, the cells, for sure. The primary antibody binds to the specific antigen, and the antibody may be, as we said, the primary could be conjugated, or we could use a secondary labeled antibody, and then the marker or the fluorescent dye. So, at first, the sample preparation. Sample preparation, fixing the sample to preserve the architecture of the collected tissue or the cells, and prepare for it for antibody labeling, permeabilization, and further steps like blocking. And sample labeling using the antibodies and visualization of immune reactions. These are the main steps. Let's look deep, have a deep look on the steps of the sample preparation, which we will discuss today in our presentation. At first, we have the tissue or the cells, and the cells will be fixed. And then there is a blocking, serum blocking, and then the antibody labeling, and then imaging and judgment. So let's take step by step. At first, we have the cell fixation. But before the cell fixation, we have to prepare the cells for the fixation step. So how we will culture the cells? Should I culture them in the plate or on the slides or on cover slip, or how? What is the most convenient or the most proper way? Actually, there is no proper way. It's all convenient and according to your experiment, but I usually, for immunizing for long-term preservation of my slides and having them if even especially if you will capture using outsource or service fluorescent microscopes in another institution, whether national or international or whatever. So I use a protocol with cells on cover slips usually, which is the traditional method with nail polish mounting. So at first I sterilize the cover slips and the slides. Sterilization of cover slips and the slides can be used. In my case, I use actually the cover slips. I put them in distilled water in a bottle that can be autoclaved, and then I will put them in autoclave for autoclave cycle, and then remove them using sterile forceps under the laminar flow, and then put them in the 6-well plate as we can see here. You can coat the cover slab with using extracellular matrix like matrigel, gelatin, especially in case of semi-attached cells like neuroblastoma cell lines. They are semi-attached, so we have, and these cover slips are not actually manufactured for cell culture. Like the plates are modified with nanomaterials and things like this, so we can use extracellular matrix for proper attachment and enhance the attachment of the cells. So we place the sterile cover slips into the 6-well plate, and then we will seal the cells in large volume. Usually I use 30K cells on the cover slips, not too much and not too little. And then after your experiment, either you do the experiment directly on the cover slips, or the cells already had the treatment or whatever the experiment you done, and then you got your cells and culture them on the cover slip, and then you will proceed with the staining or the antibody protein detection directly after putting the cells on the cover slips. So at first we will remove the media and then wash our cells using PPS or TPS washing solutions. Then here we will go for the steps that we will follow in these slides, and we'll discuss every step of these steps in details. The first step is fixation, then permeabilization using detergent or washing, then blocking, then primary antibody, then washing, then secondary antibody, then washing, then mounting. These are the procedure and the steps that we will follow. So at first, oh yeah, you can use also this chamber, which you will see the cells directly on the slide, not a cover slip, and with the chamber and do your experiment, and then remove the chamber and take the slide and do the procedure. Okay, at first the fixation. The fixation, why we will fix the cells? So we have the cells on the cover slips and it's attached and ready for protein detection and immunostaining. So the cells usually, if you didn't put any fixative on the cells, so the cells usually will go through the autolysis. So to preserve our cells and to ensure the integrity of the cell tissue and the proteins of interest to enable the antibody detection in the next step, we have to proper fixation of the cells. And the sample fixation process by which the cells are preserved, the fixation methods depend on the protein and that we want to visualize, because we have several types of fixatives. And the fixative of ideal for preserving one structure may be not suitable for another cellular structure. And the broad objective of tissue fixation, that I like this statement very much, is to preserve the cells and tissue components in a lifelike state. So the cells are not died and they're not lived, they are in lifelike state. The last process in the cells, it's fixed. Okay. In absence of fixation, as we said, the essential proteins and membrane and the intracellular structures may be dehydrated, degraded through autolysis, putrefaction, and the unexpected changes that can and the that can be done to the cells without preservation. But we have to put on our mind that under fixation or over fixation of the cells are harmful, will be harmful to the cells. So under fixation, poor morphological preservation, and can result in loss of signal. Over fixation can also cause artifacts and loss of signal, and can do non-specific binding of our antibody. So as we can see here, this is our cell starting and then moving by having the fixative, adding the fixative. There is fixation through cross-linking of the proteins together, and or coagulative fixative, and then the permeabilization, and then the fluorophore labeling. So, as the first fixative that is general and provides the best preservation of fine structures, and usually used in actually the best for electron microscope, is glutaraldehyde, and its action is through cross-linking of the proteins, and it's very good for microtubules and membranous structures, but it is harsh fixative, may sometimes negatively affect our antigenic epitopes, as we can see in the figure. And it usually we need to neutralize the points that not react with the tissue. The second fixative, which is the general fixatives used, actually is formaldehyde, and it is made freshly from paraformaldehyde. So usually we say formaldehyde, not paraformaldehyde. Formaldehyde also acts by cross-linking the proteins, resulting in fixed insoluble meshwork. It is a base for immunostaining, as I said. It is not, but it is not effective cross-linker like glutaraldehyde, as it is small in size and not long enough to form extensive cross-linking, and the cross-links can sometimes be reversible. But usually we use formaldehyde freshly formed from paraformaldehyde. Also, aldehyde, methanol, and acetone alcohols used for fixation, but we have to keep them in ice cold. So we have to keep them aliquots in the freezer to prevent their condensation. And the methanol and acetone fix actually very rapidly, and usually used for the tissues that can't undergo formaldehyde fixation, and they work by precipitating proteins. So they can disrupt sometimes the epitope structure, but they have an advantage that these organic solvents fix and permeabilize simultaneously. So they could disrupt the membranous components, but they do simultaneously fixation and permeabilization. That's why we will come to mention them again in the permeabilization step. So we have now our cells fixed and in lifelike state. So our protein of interest is inside the cells, fixed or through precipitation, coagulation, or cross-linking. The next step is our protein of interest is inside the cells. So the fluorescent dye is usually not membrane permeable. In order for the dyes and the antibodies to pass through the cell membrane, we have to form pores in the cell membrane. So we have to make permeabilization of the cell membrane to access the intracellular and the intraorganellar antigens. But if our antigen is on the cell surface or on the cell membrane surface, so permeabilization will not be done. But it's the best to do permeabilization step. So our cell membrane is phospholipid bilayer. So two general types of reagents are commonly used for permeabilization: the organic solvents like methanol and the acetone, as I mentioned before, and they do fixation and permeabilization simultaneously. And the common reagents used for the permeabilization, which are detergents like saponin, Triton 100X, Antwine, MP40, digitonin, and so on, which is solubilize the phospholipid bilayer of the cell membrane. Saponin is very gentle actually and solubilize cholesterol in the membrane, and it's less damaging. And sometimes some dyes use require saponin for the for like lipid dyes or something like that, prefer saponin, but must be present in all probe incubation steps because the membrane will reseal if the saponin is removed, because it is very gentle. Okay, the required concentrations usually of these detergents for permeabilization, the recommended concentrations are 0.1% to 0.5% of Triton, 100x dissolved in PPS or TPS, saponin 0.5%, digitonin 50 microgram per ml. Okay, the washing solutions that are usually used between every step and the next step to wash the detergent or wash the primary antibody or wash the fixatives is either TPS or BPS. Those both actually widely used for immune staining. TPS may be preferred as trace has blocking properties to mask the unbound aldehyde groups, like in we have mentioned in glutaraldehyde that some aldehyde groups are not binded, so that can trap the antibodies non-specifically. So after we done fixation and then permeabilization to enable the antibodies and the fluorophores to enter the cells, we have to make a very critical step, which is blocking the non-specific binding to avoid false positive results. So why non-specific binding can be done? Actually, due to unreacted aldehydes, like we mentioned in the previous slide, or due to highly charged or highly hydrophobic moieties that will trap antibodies, low affinity of immunoglobulins in case of polyclonal antibodies, non-specifically bind to other structures rather than the protein of interest. Can be achieved by incubating the cells with solutions containing proteins or other molecules that will bind to readily reactive non-specific sites, thereby blocking the antibody's ability to bind to these non-specific sites. So what are these molecules or solutions that we use? It include normal serum and protein solution, like bovine serum albumin, casein, gelatin, normal serum obtained from species as a secondary antibodies, like the source of the secondary antibody. Blocking efficiency evaluation can be done by performing negative control experiments in tissues known to not to express this antigen of interest, to make sure that our blocking reagent is effectively doing his action of blocking non-specific binding. The usual recommended concentration of the blocking reagents is 1 to 10% in buffer dissolved in PPS in case of bovine serum albumin or other powdered blocking molecules or reagents. So, as we can see in the figure below, that non-specific binding can be done without the blocking, but after blocking, as we can see here, per se, these are bovine serum albumin are blocking or non-specific sites to keep our antibodies bind only to the protein of interest. And the blocking step usually it takes 30 minutes to 1 hour in room temperature. And after blocking, actually we do the sample labeling. So our tissue now is ready for the antibody labeling. So to introduce the antibody to the tissue to bind to the protein of interest or our target. Primary antibodies used to detect the antigen, as we mentioned before, and these primary antibodies can be conjugated to fluorophore or it can't or non-conjugated, so we need to have secondary antibodies that will be conjugated to the fluorophore. So if we used the primary antibody conjugated to fluorophore, it's called direct immunofluorescence. If it is not binded already to the fluorophore, so we will use secondary antibody binded to fluorophore, it's called indirect immunofluorescence. Actually, direct immunofluorescence can cause less signal amplification. However, indirect cause signal amplification. And direct less commonly used because it's cost and time consuming, so you will have more than because it is conjugated already, so you will have more money to purchase them. However, in indirect, you can use primary antibodies, many, and one secondary antibody in your lab. So you can purchase one secondary antibody and many primary antibodies, but taking in consideration the host of the antibodies. So as we mentioned before, that non-specific binding can be done due to the polyclonality of the antibody. So the primary antibody could be monoclonal or polyclonal. Monoclonal produced by single B cell population and recognize single epitope of the protein of interest, but the polyclonal antibodies arise from multiple B cell population and can recognize multiple epitopes of the same antigen. Monoclonal are highly specific, whereas polyclonal antibodies are more resistant to change in protein conformation because they can bind to multiple epitopes. So if we wanted to ensure specificity, monoclonal antibodies are recommended. But if we have protein conformation changes, so polyclonal, and we are not sure about the conformation changes of our proteins now, so using polyclonal antibodies to detect more than epitope is advised, actually. And also we can the primary antibody can be as we said, conjugated, and in the conjugation, the marker here in immune fluorescence staining will be our fluorophore, but it can be enzyme as well, and it can like in immunohistochemistry, for example. As we can see here, monoclonal antibody binded to single epitope, and the polyclonal antibody can bind to the multiple epitopes of the single antigen. False negative results can be caused by why we can false negative results we can have in our tissue or our samples due to a conformation change induced by fixation, low affinity of the antibody to the target, failure of the antibody to penetrate into the cells. So these all can fail our primary antibody to detect their target for antigen, even if it is in the tissue. And false negatives can be caused by this primary antibody bind to another antigen. Visualization. So we now introduce the primary antibody, either it's conjugated or non-conjugated, and we will have then the secondary antibody. Regarding incubation of the primary antibodies with the cells, actually, you will follow the protocol of your antibody, actually. Recommended protocol for each antibody, but usually it's 1 hour in room temperature or overnight in 4 degree, and avoid the over incubation of the antibodies with the cells to avoid non-specific binding. And you will follow the recommended protocol for each antibody. And the antibodies, usually it is diluted in 1% antibody dilution buffer that contains actually bovine serum albumin, for example, or normal serum. For visualization, This step includes the interaction between conjugated antibodies with a primary or secondary one and the protein or the protein antibody complex. Causes a signal molecule that are attached to the antibody to either excites, in case of our immune fluorescence, excites the fluorophore at a specific wavelength, and it will emit light at another wavelength. So the excitation in a wavelength and the emission in another wavelength that will be detected using the fluorescent microscope, or emit a signal that converts undetectable substrate into a visual signal, like what is done in the Western blot technique. At the second labeling incubation step, usually the antibodies are labeled with fluorescently labeled secondary antibody, but we can also use non-immunological fluorescent labeling agents. What is the meaning of non-immunological fluorescent labeling agents? Non-immunologically fluorescent labeling agents like DAPI, Hoist, or phalloidin, they are serves as important counter stains for non-biological structure, which allows you to determine if a protein of interest is associated with the structure. And they usually and they are not antibody based. They are stains that bind to specific non-biological structure inside our cells. So there is no the step of the first primary antibody blocking or secondary. There is no antibody needed for these non-immunological fluorescent labeling reagents. Phalloidin, for example, they can label specific for the actin that be used for the double-stranded DNA and not ideal for live cells because it requires permeabilization of the cell membrane. In contrast, Hoist labels the DNA is lipophilic and it can permeabilize the cell membrane, so it can stain the live cells because fixation permeabilization steps are not needed in while using Hoist. So if you want to label the DNA while your cells are in culture and they are living, so Hoist is recommended. But if you want to, if you are going to fix and permeabilize your cells, so that can be done as well. So Hoist can be live or die or fix it. The right term is to fix itself. DABI is ideal for DABI only for fixed cells. As we can see here, this is a PKH26 dye that labels the cytoplasm of our cells here, and this is the Hoist. The cells are actually is viable, as we can see. These are examples of non-immunological fluorescent dyes, and we can see our cells are attached, living in our cell culture, and we stand them using, I stand them in my lab using the PKH26 dye for the cytoplasm and Hoist for the nucleus. So here comes the fluorescent reporters. We are repeating that our antibodies labeled by fluorescent dye, fluorophore. What are fluorescent reporters? Organic fluorophores or fluorescent dyes such as fluorescein, isothiocyanate, FITC green that gives the green color, or rhodamine red color, are conjugated to the primary or secondary and frequently used in ICC. Immunocytochemistry or immunostaining or immunofluorescence staining. Fluorescent reporters don't require substrate to activate an enzyme, and high resolution fluorescence microscopy can be used to detect the fluorescent reporters' existence. And their advantage over the other reporters like enzymes or radioactive materials that multiple signals be visual, so you can visualize multiple targets proteins at the same time simultaneously. But like primary antibody against actin, for example, and using secondary this primary antibody, for example, a rapid antibody, so the secondary will be rapid as well. And another antibody co-incubation with detecting another protein like TP50, B53 cancer protein, and but it is mouse antibody, and the secondary antibody will be specific to this primary antibody, so secondary antibody mouse as well. So but we, in case of multiple antigens, so the fluorophore attached to the rapid antibody for detection of the actin, and the fluorophore attached to or bind to the antibody for detecting B53 should be different fluorophores or different, giving different colors. Here red color, for example, and here a green color FITSI. So you have to choose your, in case of multiple antigens to be visualized simultaneously, to choose different hosts of the antibody, anti-mouse with the anti-rapid, or anti-goat with anti-rapid or anti-mouse, and so on. And also you should take care that you choose different fluorophores, different colors. So each antibody gives a different fluorophore. As we can see here in human colon carcinoma section, for staining the cytokeratin 18. It is cytokeratin 18 is labeled with red, the antibody red fluorophore, and the blue color for the nucleus. Fluorophores, as we mentioned before, producing the various colors, and the excitation is at the wavelengths, and the emission in a different wavelengths. Like Alexafluor 488, the excitation happens at 499. However, the emission at 520. Okay. Okay, now we stained our cells with the antibody labeled to fluorophore. What we will do next? It can be dried by the environment, by the air in the environment. So we have to keep our cells. We have to avoid photo bleaching of the fluorophore signal, and avoid shrinkage or drying of our samples, and keep them for long term, uh, weeks or months even. But with time, photo bleaching will be done for sure. But it can be kept for a long time. I kept them for months, and it was okay. Uh, Here comes our hero, is the mounting medium. Mounting medium is a medium that your sample is in while it is being imaged or in the microscope. So we put like one drop of if we cultured our cells on cover slab, so we will have a slide, and this slide we will put a drop of our mounting medium on that slide, and then put the cover slab on the from the face of the cells to between, so the cells will be between, as we can see here. This is a standard glass with mounting medium drop on the standard glass, and the cover slabs will be converted and put on the slide with the cells in the mounting medium between the cover slabs and the standard glass. So what is the importance of mounting media, and why we use mounting media? There are several reasons to plate them. To help hold the specimen in place while they are imaging, to prevent your sample from drying, to this mounting media actually has the same refractive index of the cover slip and the glass slide, prevent photobleaching, preserve your sample over time for long-term storage, as I mentioned. So all these are the importance of the mounting media. Types of mounting media can be solidifying media or non-solidifying medium, like glycerol-based mounting medium. Always, after putting the mounting media and having your cover slip, there is actually important step, which is seal your cover slip with nail polish, especially in case of non-solidifying mounting media. So you, on the four boundaries of the cover slip, you put nail polish, which is a transparent one, any nail polish transparent, just to make sure that the cover slip well sealed to the slide and will not be moved from your slide, and let them dry in air for sure, because once you put incubated your sample with the fluorescent antibody, you have to work in dark to avoid bleaching, photo bleaching of our fluorescent dyes. And always image through the cover slip, not the slide, because of the thickness of the cover slip. Cover slips are thin than the slides, and also the cells are where if you remember the first step, were attached or adhered to the cover slip. So you image from the side of the cells to see for proper visualization of your labeled proteins. Slides with solidified mounting medium can be kept for at least 1 to 2 weeks at 4 degrees. I usually keep them at 4 degrees for short term, and for 1 month at -20. Samples prepared with glycerol media will not last as long as the solidifying mounting mediums. Make sure while you are using the cover slip to choose the right cover slips for your objective and of your microscope. So cover slips comes in various thickness, like 0.08 or 0.03 to 0.17 millimeter, and so on. So in case here you will you should actually you should take care of the numbers on the objective lens, which is the numerical aperture number 0.17, 160 over 0.16, sorry, 0.17. What is the meaning of this? Actually, 0.17 refers that this is the thickness of the cover slips proper to be corrected using this objective by the designer. So the designer of the objective indicated that the thickness of the cover slip should be 0.17 to be corrected by the objective lens, this objective lens. So you have to take care of the thickness of the cover slips that is used. And eventually we'll image using fluorescence microscopes. There's a light source excitation, as we mentioned, at certain wavelengths specified for each fluorophore, and will be emitted in another wavelength and detected using the fluorescent microscope. Keep in your mind that immunocytochemistry is like cooking. There are many recipes out there, but some of them don't work out very well. However, when they do, they are great. Like Professor Krohn's we mentioned at the first of our presentation today, you need the right ingredients, a dose of experience, and a few tricks from all the cooks, and a grain of common sense to have pretty, really very pretty images and detect your protein. And thank you. I would like to have to know if there are questions or.
Now, it doesn't seem like there's any questions in the chat. So I think we can close it off here. As Nehal said, you guys can please email her Nehal, will you please put your email in the general chat? Please do feel free to contact Nehal if you have any questions about this presentation. We will make sure that it's available on YouTube as well, if you would like to watch it again at a later stage. But thank you all so much for attending, Nehal. Thank you so much for this informative presentation. It is so helpful. I hope we will be hearing from you again about something else maybe in future. But this was very informative and very helpful. I'm sure you'll get a lot of emails inquiring into your expertise.
Okay, thank you, Precious, and thanks for everyone attended today. And thank you.
Thank you, everyone. Have a good afternoon.